Hello, Mr. Allman here, and this is your Allmancast on cell transport processes. Now, a related concept is the idea of tonicity. So tonicity is going to be the relative measure of solute concentration. How much solute is there inside versus outside of any one particular cell? And its tonicity has important consequences on the health and well-being of that cell. So if you look at each of these situations, you should be able to explain based on osmosis what is happening. Remember, osmosis is going to be the movement of water towards a higher solute concentration. Now, if a cell is in a hypotonic solution, that means that there is more solute inside the cell and less solute outside of the cell. So water is going to move towards where there is more solute. It moves into the cell, it fills up the cell with water, and if there isn't a cell wall present, that could cause the cell to burst. And that's called the process of lysing. A cell that is lysed has been filled up with water to the point that it explodes, much like an overfilled balloon. Plant cells have a cell wall, which is going to help protect the cell from exploding. So plant cells are actually well conditioned with that cell wall to prevent against that process from occurring. In an isotonic solution, there is an equal concentration of movement into and out of water into and out of the cell. So a normal blood cell should exist in an isotonic solution. There should be dissolved things inside the blood cell, and there should be an equal amount of dissolved solutes in the blood plasma that surrounds it. Now, if it is a hypertonic solution, now there is more dissolved stuff outside of the cell. And that's going to draw water out of the cell and into the surrounding environment, because water will move naturally towards an area of higher solute concentration. So in this case, the blood cell is going to shrivel up as it's dried up and water is drawn out of the cell. Likewise, water is drawn out of the plant cell, and in these cases, the plant is going to become flaccid. The leaves are going to droop, the stem is going to bend, because the cells in that plant are no longer stiff and upright. And so having that hypotonic solution is really important for a plant in being able to maintain its rigidity. Now, the movement of water and the movement of solutes doesn't depend on any one particular solute. It depends rather on the total dissolved substances. So if you look here, M is an abbreviation for molarity, which is a measurement of concentration. Now if you add up the total dissolved solutes within this dialysis bag, you would find that 0.03 plus 0.02 gives a total of 0.05 molar solute concentration inside of the cell. Conversely, you have 0.01 molar sucrose, glucose, and fructose in the surrounding environment outside of this dialysis bag, which gives a total concentration of 0.03 molar. So 0.03 molar. So you have to add up and consider the combination. So when you combine them, you see there is a higher solute concentration inside the bag. Therefore, water is going to move from where it is less concentrated outside and into the cell. So now you can see that this cell is in a hypotonic solution, and water is going to move in and cause an increase in the total weight of this model cell as water fills up the bag, assuming that those sugar molecules cannot move across that semi-permeable membrane. Finally, on the issue of osmosis, we can apply a little bit of knowledge from the paramecium, which you learned about previously in the unit. The paramecium does not have a cell wall, which means that it is subject to osmosis. Now, these cells usually live in a hypotonic solution, which means that water is going to be consistently moving into and filling up that paramecium. Now, the paramecium, if it bursts, dies. So it needs some way to be able to deal with that excess water. So it has an organelle called a contractile vacuole, which here you can see is fairly small. But as the cell fills up with water from its environment, that contractile vacuole you can see now gets much larger. And once that contractile vacuole is filled, that vacuole is going to merge with the plasma membrane and expel or release all of that water out of the cell. So in other words, the bag fills up, and once it's full, that water is ejected. And that process needs to happen continuously to ensure that the paramecium doesn't fill up and grow too big in size and burst. So just to summarize here, you can see the difference between passive versus active transport. Passive transport is always the movement from a greater to a lower concentration. In all cases, passive transport does not require energy. It moves by diffusion. It could be simple diffusion, in which a small nonpolar molecule moves directly through the phospholipid bilayer, or it could be a type of facilitated diffusion when a transport protein is used to be able to move that molecule across. Active transport is used in the opposite case when a molecule wants to be concentrated on one side of the membrane. So this is in the opposite direction of diffusion, and ATP or some other source of energy is required to move that molecule against its concentration gradient. Finally, if things are really, really, really big, transport proteins are not big enough to be able to get them across. So we have a process to be able to bring in and to get rid of those materials. Endocytosis is going to be a process describing several ways in which large molecules are brought in. Phagocytosis is going to be a method by which a cell is going to eat or bring in a large nutrient molecule, or in the case of something like an amoeba or paramecium, a whole smaller bacteria cell. The plasma membrane is going to extend and surround that food particle, and then it's going to entrap that food particle within a food vacuole, which may then be fused with something like a lysosome that has digestive enzymes that will break down and allow for those nutrients to be used by the cell, by the cell's mitochondria. Penocytosis, or cell drinking, is a process by which small molecules are going to be brought in and trapped within small vesicles. So here we're dealing with individual molecules, not one really large piece of food or nutrient, but many smaller molecules. In these cases, we have very small vesicles, very tiny little things containing only droplets, small collections of individual molecules. And finally, receptor-mediated endocytosis is when a particular food particle or a particular signaling molecule is being looked for and brought in. So there are receptors on the cell membrane, and if and only if a matching molecule binds to those receptors, will it bring it in. So this is much more specialized, in which a part of the membrane is looking for one particular type of molecule. Then finally, exocytosis is going to be the release of a large cellular product. So maybe, for example, a group of proteins, such as the hormone molecules produced by the alpha cells or beta cells, the beta cells of the pancreas. So in this case, we have proteins being made on the rough endoplasmic reticulum, being sent out by a vesicle, merging with the Golgi apparatus. The Golgi apparatus then modifying, changing the shape, or in some other way signaling the release of that molecule from the cell. So once those changes or modifications have been made, a new vesicle is formed, which pinches off of the end of the Golgi apparatus, and then is going to be moved along the cytoskeleton to the plasma membrane, where it will merge, and after it merges, it's going to then release those large molecules to the exterior of the cell. That vesicle, what the phospholipids in that vesicle are now going to merge with and become a part of that plasma membrane. The process there is important. We can see this bulk transport being exemplified here in a phagocyte. A phagocyte is a type of white blood cell that's responsible for finding and killing pathogens that are within your body. And you can see here the extensions of the plasma membrane that are literally going out and grabbing bacteria, surrounding those bacteria, bringing them in so that enzymes on the inside of this white blood cell can then digest and therefore kill that bacteria, eliminating the threat to you. So this process is really important for maintaining your health, fighting off disease. As always, if you have any questions, please let me know. Thank you for listening.